All right. Well, hello, everyone, and welcome to the ChUG podcast. I'm the host, David Leclerc. I am here with Pat Sims, Ryan Duggan, and Laura Johnston. Hello, people.
Hey.
Hey.
Hello.
We don't have a topic today. Roughly a year ago, I think we passed our anniversary date, but the four of us were together to kick off the very first ChUG podcast and decided to do the exact same thing. We don't have a specific topic of discussion today. We just wanted to get back in touch and see what's going on in the world of flow cytometry. How have you guys been doing? Pat, what's going on?
Well, we had, it's been an interesting year. The biggest challenge is I've been alone for 18 months and during the pandemic and right immediately afterwards, that was okay, but it's been kind of a difficulty because that's a lot of work. So we've been trying to get people, I think our salaries are not quite in range with what people expect. I see that I've only gotten a few applications, but those expectations are way out of range with what we're offering. So that's kind of a problem. And there's just not a lot of help available. I don't even know if there's entry level, but certainly people with experience are not readily available. And Balaji from the University of Chicago is experiencing the same thing. We made a guarantee that if we got somebody and had another one, we would start sharing them because it's just been a dry well that we have been trying to get water out of. So
it sounds like.
What's your typical staff size?
My staff size, 2 to 3, and I'm 0. It's just 1 now. So that's been hard. And then I had mentioned earlier that we just discovered that our water source has algae in it. So I have algae in the Fortessa now. So I've got to take this instrument down and I've got to try to decontaminate it because it's rather expensive to have BD come in and replumb the whole thing. So I'm going to try to do that, but it's hard to squeeze that into the already overloaded day that I have. So I want to whine about that a little bit. I hope I was successful, but it's just a challenge because I've always wanted to have excellence in the facility, and obviously a algae-filled Fortessa is not the characteristic of an excellent facility. So it's just a struggle.
It is. I think it's beyond the field of cytometry, but like there's a lack of. It's many people offering jobs and very few people finding candidates. And
well, how is advertising positions these days? I mean, I remember at U Chicago there wasn't really much of a effort to advertise, but I know like whenever there's positions open at AbbVie, it's like everybody's LinkedIn page turns to the advertisement of the position. Everybody kind of like shares the same thing. I mean, I guess there's also, the route of an actual headhunter, but I mean, for kind of the entry or level tech, it's probably not really the way to go. But I don't know, what else do you guys do in terms of trying to get the job opportunity in front of more people?
Well, most of the like Monster and all of those other job sites actually sniff them out. And right. So that's where we get it. And I put it on the Cytometry group and I put it on LinkedIn. And what I get is a lot of people who want visas. That's of my 8 applicants, 6 are people who want visas.
Do you find out where people heard about the job?
I think it's on the application. I don't know that I really looked at it. I'll go back and check that out.
Wonder if it's partially the universities too, because I remember ages ago, right after college, when I was applying for entry-level laboratory technician jobs, and I had applied to the University of Washington, so very gigantic university, and they, funnel all of their applications through the same spot in the university. And I was told that the university's application process, they like filter through all of the applications. And so if your application has certain keywords in it, then it will get passed on to the person. But if not, then the person might not even see it unless the hiring person thinks to request to specifically not have filtered applications, and then they personally have to filter all of them. And I remember I didn't start getting responses until I actually talked to somebody who worked at the University of Washington, and I told them like the exact job numbers I had applied for, and then they went to HR and like told HR to pull my applications and send them to the person. So it might be some sort of like university process that's also making things challenging, I wonder.
Well, I already called HR and told them, I'm not getting anybody for these applications, and they had some ideas to boost the notice out to more people, but that wasn't talked about. So whether that happens or not, I don't know. At least at our place.
Yeah,
I'm not sure how.
I'm sure all the universities are different too.
Right, but that's a good idea. I can now check out and find out if that's it. And the other part of the problem is that, and I don't know if you've faced the same thing, but and Johnny Moore used to talk about this, that the HR departments who figure out the compensation and the compensation committee don't really understand when you put in there the job description, that it's a lot more than what that job description actually says. The knowledge that makes you able to do those functions is hard won, and you have to actually spend some time and energy to get to the point to be able to fulfill those. But the compensation committee looked at mine and they said, well, you can't offer any more money. These aren't really big skills. And I remember Johnny Moore saying that she had gone to fight with her HR department, and she actually had them structure the flow cytometry facility, their salary structure, to be in accord with the IT structure. And because IT gets paid a lot for their time and for their education, because that's something that seems to be valued more than the work that we do in the laboratory.
Yeah.
So
we talk about developing this job ladder for core facility people, and you may argue that it's not great skills, but it's huge responsibilities. You're working with somebody else's experiments, and with very limited amount of information to begin with on these experiments. So it is quite a bit of to put on the same person. I don't know.
Well, and also, if you hire somebody, I had Ashley who worked for me, and she was stupendous, but she was a recent college graduate. So everything she knew, she learned in the core facility, but she had never isolated PBMCs. She had never stained cells. She'd never picked up a mouse. So there was a lot of stuff that really goes into making you a good core person that's not actually being in the core.
Right.
Yeah. So, get what you pay for.
Yeah. Ryan, how's life in AbbVie, in industry?
Good as always. Yeah, I mean, I guess the one thing that appears to be somewhat different is like the back to work post-COVID scenario. I've been in the lab most of the time for the past 18 months or whatever. But yeah, I'm hearing like more and more people getting back to it, and now there's even discussions of like, what does flexibility look like now in the workplace? Do we build that in specifically? Do we just allow it to happen kind of at the manager, individual team manager level? So, for example, if like in the flow core, I can kind of manage amongst the three of us that are in there who needs to be off, if kids are sick, or, if, the school's closed, we have to go back to, daycare fiascos again. I'm more of the mind of let's make that happen at this low level within a team, as opposed to maybe some blanket statements saying this is the way things have to be done. But it was weird in general to have proposals like, oh, well, maybe you're at home 2 days a week and in the office 3 days a week, as opposed to kind of what I would prefer is kind of just a flexibility of, yeah, I need to come in to run an experiment, great, but I'm going to be done by 2:30 or something like that. So then I'm going to leave and analyze data at home or something, or read papers at home or whatever that is. But I don't know if that's happening all over the place or if that's something unique. But I wonder. Yeah, I'm sure it's also regionally different,
Right.
right? I know some folks at universities on the West Coast, even in core facilities. I mean, they are minimally in the lab still.
Yeah.
Right. And so they're even like, let's plan our sorts on Mondays and Tuesdays or something like that. So the sort operator only has to go in at those days at those times and whatnot. So I think it's, yeah, I mean, I feel like at least in my little part of the world, things are pretty much back to normal. I mean, we're all present, working, everybody's doing experiments.
Vaccinated.
The one thing I will say though, that is still held over is the lack of supplies.
Yes.
Oh, really?
Of like we still struggle to get pipette tips and the specific plates we like to use on the Aurora and the Fortessa. We've had 96 well plates. I like these V bottom polypropylene plates that I probably get from VWR or something like that. But they've been on back order since like last summer. So over a year, we have them on order. They just haven't come in yet.
Wow.
So obviously we're using different plates, but that and like pipette tips. I mean, I do typically some studies are with, 40, 50 mice, 3-4 tissues per mice, multiple panels. You go through a lot of tips, right, to do all that kind of staining. And man, I feel like we're just blowing through tips and we just have nothing in reserve. It's always like we're actually getting to the point where we're bartering with other labs saying like, oh, we'll give you some of our pipettes if you can give us some pipette tips and things like that.
We're okay with Mike ordered a bunch, like a ton of bags of 200 micron tip, but in bulk, in bags.
Ooh.
And we hated like having to like fill those, so we just left them in the back.
He said that he was okay filling all the
Yeah.
individual tips himself.
Absolutely.
I don't know if he's done that, but he said he would take responsibility for filling them individually.
Anyway,
Yeah.
so now I found value in those bags now.
Yeah.
Yeah. Yeah.
Wasn't
Well, maybe
there
while he's sitting for hours on hours at the CYTO, you can feel
Wasn't there a glove shortage recently for us? Wasn't there? I heard something about like.
Yeah, we're back ordered on gloves too. I mean, you can get gloves, right? It's just like everybody has their own special kind that they want, and yeah, we have like a variety pack of different colors and styles and materials because that's all we can get.
I think we also we definitely in February had a shortage on flow tubes with caps, which was a bummer.
Yeah.
Especially when I was trying to work with samples that needed some biosafety stuff. So I wanted caps and I couldn't get them.
Laura, what's new in your world?
I guess recently I've taken a deep dive into YouTube training videos. So I've
making them or watching them?
Making
Oh, okay.
Making flow cytometry YouTube videos. They've been like slowly increasing in quality over time. And the most recent batch that I made, which was redoing my Aurora training course, I had recorded, it was the first time I had recorded bits and pieces at different times. And so by the time I spliced all the video together, the volume was different levels. And so I needed fancy video editing to normalize the volume. So then I went into this whole thing. So now I'm skilled, well, maybe not skilled, but I have knowledge now on more video editing techniques, which is kind of an interesting thing that's come out of this pandemic. I don't know if I would have really spent as much time making videos.
Absolutely not.
But now I kind of think they're fun. So
Yeah,
then
I kind of want to tie that in with the, so a bit, as I said before, a year ago, we started the podcast, and that's totally a pandemic thing. I think ChUG would otherwise have tried to maintain a quarterly in-person meeting with some kind of presenter and pizza and beers, and like that would have been it. But instead, we started doing first webinars. We had Mr. Tim from Flojo.
Crawford.
Yes,
Tim
Tim Crawford. And David Novo came afterwards to explain on mixing. And then I kind of got annoyed at the idea of like having live event at a specific time, making sure people show up. And so we just decided we'll just record podcasts and people can watch that whenever they want. But all these skills that we develop over the year are just totally due to that pandemic. I probably would not have bothered with any of this otherwise.
And I don't know if it's a function of laziness or convenience or what, but there is something really nice to knowing that we had this meeting 7:30 to 10 tonight, but I could be home.
Yeah.
You know, there is something really nice about that. And it makes me more actually wanting to do things than, you know, so that's I think the double-edged sword, because I miss the in-person part of it as well. But yeah.
Yeah.
Yeah, I think it's made our Aurora training stuff more successful in a sense. Like, I don't know if I would have necessarily thought to make so many YouTube videos, but now that I've made them and I know I'm capable of it, then anytime some complicated topic comes up in regards to Aurora training, I just make a video. Like, I made a 10-minute video to explain to people, now that we have more than one Aurora, how should you plan out your experiments? Because it's kind of complicated if you want to run an experiment and combine it in with the second one, and we have two Auroras and they're kind of the same, but they're not exactly the same. And I'm like, I'm just going to make a video to explain this, because otherwise I definitely don't want to type it up in an email. That's going to be a novel.
And nobody's going to read it.
Yeah, but I'm pretty sure we definitely have views on that video, so
Oh, good.
makes sense to people.
And you're not reinventing the wheel each time. You know, I still give the FACS class, the introductory FACS class, and it's the same thing every time, and I'm bored to death with it. But I've been teaching it for a very long time, so you know, that's a good idea. I like that.
Yeah, I just have a personal interest in the training stuff, I guess. And also I like making PowerPoint slides look pretty. I could spend forever making PowerPoint slides look pretty. And so I'm always just kind of like, could I make this better? Could I explain it better? Could I have a better graphic for this? And it's also a perfectionist thing, which is, I don't know, good and bad.
Yeah.
Okay, so I just went to the UChicago Flow YouTube page.
All right.
There are, quite a few videos here. Well done. I'm going to have to check these out.
Yeah, I have all kinds of videos. I have
And I'm going to
longer form,
subscribe.
shorter form.
And I'm going to subscribe.
And like.
Yeah, right.
The other part is learning how, like, what people like and what you like to watch. I know I've done a 30-minute walk through the Diva software, which I can't watch anymore because it's so long. It's so like, no, like I'm taking the other softwares. We have we go through the software that runs the Attune. We now have a Quanteon. We're gonna go through that as well. I can do that in 5 minutes. That's going to be enough. And then if you want to cover specific topics, just make more videos.
Yeah, what is that? The sweet spot is somewhere like 7 to 10 minutes or something like that.
Yeah.
Except for podcasts, right? Because, you know, when people are watching podcasts, they know they're in it for the long haul.
Well, David pointed out on YouTube, they have this chapter feature.
Oh,
So
yeah.
you can actually break down. So now all of our newer videos, I go through and find the actual time where I start talking about certain subjects or like doing certain things in the tutorial, and we break it down in chapters. So if you just want to watch the one section of the 30-minute video, you could just go directly to that one, which is nice.
Very good. Wow.
What's new in my world is I've been dealing with the aftermath of a flood, which I think there's many floods in history. Talked about the biblical one, and then that time Atlantis fell into the ocean, and then just below that I think is the GET facility flood, where this little piece of tubing cracked. And then water started spewing straight up to the ceiling, which is a tiny amount of water, but like over time, it's surprising high amount. So it started spraying all over the place, and the ceiling got soaked, and all the tiles started falling down on the instruments, which after a while, it's like basically it's kind of a giant sponge on top of each machine, and then the water just like, there you go. These are, pictures
man.
Wait, so it was a pipe that broke?
You know, the pipe that brings the
No, it
water
was
to the
not.
milk
It was
machine.
not that. That happened to us before.
Yes.
That was the only source of
The
water.
month that you left, Ryan, that's the exact same problem.
Because remember, that pipe broke and it was all over the floor, and Sasha Shervansky called me up and said, "Hey, your lab is flooded." I'm like, "What?"
Yeah.
It's the same thing.
Yeah.
Oh
We
man.
explained, for if you're not watching on YouTube, we're showing pictures on YouTube.
Yes.
If you're listening to us, you obviously cannot see the pictures.
But it's basically a small tube spraying small amount of water, but at a really high pressure.
Yeah.
And
Wow.
it just so happened that leak started in the middle of the night on a Saturday, and then the next users were midday on Sunday, and then I got a call at 11 from Dr. Sperling, who is my boss, saying like, should probably go back to the lab. There's a flood, the machines are destroyed and whatnot. And then it took me 2 hours to get there. The water didn't get cut off until like maybe half an hour before I showed up on the.
Oh my
Oh
gosh.
man.
That's the problem, was we couldn't get the water shut off. But
Right.
that little, so if you, this part here, this is
Yeah.
behind the millicue, right? So the millicue is right here. So the water source is like behind. And if you look at the ceiling tiles, the trajectory makes sense.
Yeah.
Even though it's a really tiny amount of water. I mean, I wasn't there to see it spraying, but like the trajectory of the ceiling tiles, it makes sense that it's coming from that one
Wow.
spot.
Yeah.
So
Wow.
what's so the two auroras
All right,
Yeah.
there.
hit, and what's on the other side of the wall?
The Fortessa X20 and the Attune. So this is the bunch of drawers that got
Oh,
filled
man.
with water.
Drawers filled with.
So we
Oh,
lost rolls of tapes and tips, and the Meso Quickplex SQ21 manual,
Critical.
which like
Critical to have.
we're screwed now. We need the. I have no idea how to use this thing.
So did the amnest survive?
Yes.
The amnest was not touched.
It's
Wow.
right here. It just missed it.
Yeah.
That's the amnest. This is one of the auroras. This is the second aurora. And then
So
on
right, the one thing that I thought was interesting is, so after that we started looking at the damage, right? And the X20 has a fan right on top of the top panel, and the tiles, one tile fell right on top of it, so water started dripping completely inside the machine, right? X20 has a cover on top, so it's harder for the water to get inside, but like it kind of finds a way on the sides of the machines, and in the back of the instrument there starts to have holes that connect to the different boards. The Attune is the other machine that got hit by the water, and this one was covered in water, but this is the X20s with the tile on top. The Attune is kind of built so that the water just does not get inside and does not get in touch with the electronic boards. I was blown away. And so like initially I kind of like, I thought all the instruments were destroyed. And then looking at the Attune, I could not find a single trace of water on anything remotely of interest for that purpose.
Oh.
Well,
Laura's trying to push a picture of the
oh, there we go. You can get rid of the Fortessa anyways, right? I mean, that's been around for
it's
a while.
2016 we got it.
Yeah.
So
who wants to use a Fortessa anyways?
Come again?
Who wants to use a Fortessa anyways? Just kidding.
The Attune, I think we have to replace the plate loader unit. It has a name, I forget what it is. But the Attune itself is totally fine.
How's that monitor?
Not so good.
Yeah, that sounds pretty terrible.
Yeah, absolutely.
But yeah, I guess with the benchtop analyzers, we had some satellite facilities that weren't normally at full capacity, and so a lot of things have been transferred there. And I guess we got a Quanteon so we got a brand new analyzer in the meantime that arrived, so that's fun.
Can I ask how you're getting these analyzers? Are you guys writing shared instrument grants or this cancer center stuff or what?
So we have a 5-year plan that we made the OSRF aware of. And we're basically explaining, okay, so for example, we had this LSR2, it's gonna, the support ends in 2022 and so on. So well, all these things are planned and budgeted. And so when the year comes where we said we're gonna ask an instrument, we ask for it, and typically there's no, not that much quarrel. And every now and then, for example, the Quanteon was purchased by a new venture at the university. It's the Jonas Center for Cellular Therapy. And they got a giant budget to get started their thing. So, and it's Dr. Schreiber was my partner in getting that instrument. We have a pretty good relationship. And so he knew he wanted to replace the LSR2 that was in that satellite facility, but didn't want to really deal with, like, you know, the day-to-day management of the instrument. And so he said, well, we'll give it to you whenever it breaks, you fix it. And then essentially they purchased the instrument. We gave them access, priority access in the afternoon, and then so that's basically the two main ways, I think, at the university we're finding new equipment. Otherwise,
Yeah.
I think NIH would not support our request all that much anymore. We're just too big.
Such a reasonable faculty member you have there.
I know.
Oh,
It's I
pretty
think
funny.
it's a very good case that most PIs would agree. Like you don't want your own instrument. You don't want to pay for lasers when they break. You don't want to. You want the training. You want to pay for training of by staff that is expert in the field. So there's two groups like that we were able to federalize in a way. So where we developed this partnership where they use the instrument and we get to charge based on how they're used, and then we take care of pretty much training, repairs, all these things.
Yeah, that's a great model. I mean,
Yeah.
It's a good deal.
Yeah. And how are you liking that Quanteon?
Love it.
Josh bought it.
Me too.
Brand new.
Yeah, we've had ours for a little over a year, maybe a year and a half. Yeah, it's a workhorse.
Right.
So reliable. Like my favorite part about, I don't know if you guys have like the sample loader that does everything, right? So that's probably my favorite part. Like tubes, any kind of plate, set up a rack of 40 tubes. I mean, you can kind of pretend like you're doing high throughput, but still have the comfort of working in tubes still.
Yep.
But yeah, I'd say like the sample interface, kind of all those neat little startup shutdown tricks that they do, ease of doing those types of tasks. Yeah, I mean, whenever we have a new user coming to want to do flow, we automatically put them on the Quanteon.
Mm-hmm.
Like we have a Canto, we have a Fortessa, we have the Auroras, but it's like everybody, unless you need to do a bazillion colors, you're going to use the Quanteon, and that works out great.
I think Laura had, because we're also thinking about getting a Pantheon down the line, and Laura had questions about whether it is. So the issue is eventually, so on these machines, the voltage on the detectors is said to be fixed, or more or less, it's out of the box, it's optimized to get you the best separation. I think they do that on their 8-peak beads. They find the voltage that provides you the best separation on these on these beads, and they go, okay, go with that, and you're going to be fine. And the question is, once you start adding on the Pantheon, you can have there's 30 parameters available in your panel. Is there a point where you would actually want to be able to mess around with these voltage and find a way to reduce the amount of not the spectral overlap, I want to say the
Spreading error.
Spreading error. So in your experience, Ryan, is that something that you find the need to do, or?
Well, I'd say you can run it like any other instrument. I mean, right. If you want to go through and do voltage titrations and find your own set of settings, great. I mean, you can. This is just, yeah. So ideally, this is giving you a head start. And so if you actually calculate the usable log decades on, say, the Quanteon versus, say, the Fortessa, there are, in fact, more usable positive log decades on a Quanteon. So I can calculate it about like just over 4, it's like 4.1. And some of the channels, like on the Fortessa, it'll be 2.5, maybe up to 3.5 at best. So there is more room on the upper end of a Quanteon scale, so that the chance of things being too bright is diminished. And that's kind of the main reason you would want to start messing around with voltages. The other reason is, like you guys are mentioning, is to try and optimize for any kind of spreading in a high dimensional panel. We don't do very many high dimensional panels on the Quanteon. I mean, I'd say probably the most colors somebody has done on there is maybe 6 or 8 or something like that. In that case, you know, they're so spread out across the spectrum that they don't have any compensation issues really. You know, you can pretty much get 6 on a 4 laser system and not have to worry about any kind of spread. But I would imagine if you wanted to, so I even bought my Quanteon and I took detectors out. So I think the Quanteon comes with how many detectors, like 20 or 22 or something like that?
Depends on your model. We went with a 16, but then you can like, yeah.
Yeah. So when they came out, they were offering a 4 laser, I want to say it was 22 channels or something like that. So, but if you look at the channels, you know, at least 6 or 7 of them are completely useless. So they're like, before, say, the Nova floors came out, that kind of filled some of those gaps. So when we purchased it, like, yeah, I cut it down to, I think maybe 16 fluorescent channels anyways, which then kind of guided the end user to say, okay, here are the 16 colors at your disposal. But again, you know, most of the time people are running 2 and 3 colors on it, but you know, people might run as many as 6, which is perfect. You know, perfect setup for that. And then we tell them, don't change the gains. Unless, for example, like CFSE is way off scale or something like that, and you have to, you know, then it's more of a, you know, come get us and we'll help you adjust the gains, because, you know, you're going to get yourself in trouble if you start putting the gains down way too low. But yeah, I mean, for the most part, it's worked out. We've run it at kind of the default settings for 99% of the applications that we've run on there. Again, we don't do any high dimensional stuff there, so it's not really an issue. But yeah, it seems to be working out.
I guess it depends on how, I feel like people set their gains or voltages differently, and there's like different perspectives of how you could set them, because
people like companies,
no, like
or
you
individuals,
researchers
okay,
running experiments. Because I was trained by immunologists, and I hadn't really talked to that many flow cytometry expert people until joining the Corps. And so I hadn't really heard of all the voltration tests and, you know, finding the most optimal setting for each detector. I hadn't heard of that method until joining the Corps. And so I was instead taught to do basically how SciTech had set up their voltages. So, you know, if you think of all of the detectors and you're running the PE single stain control, then you want your highest signal to be in the PE, the detector that you've assigned to PE, and then all other spillover detectors are less signal than that one. So that's how I've always set up voltages for years and years and years, and that's what makes sense to me. And so then when I asked, when we got the Quanteon installed, they don't necessarily adjust for which signal is highest in which detector. They just find what setting captures all of the beads. So I saw some data from somebody who had come from another university to ask for my consultation services, and they had run on a Quanteon, and it seemed like when they had run on the default Quanteon settings for one color that didn't have that setup where it was highest, like I forget what it was. I think it was BB711 and AF700. I think when they ran the BB711 control, the signal was actually higher in the detector that was assigned to AF700 than BB711. So I think the data actually kind of looked okay. I think it was, I forget how many colors. It was over 10 colors. It was a reasonably sized panel. It kind of looked okay, but it just was not how I was used to setting things up. So it kind of threw me off. But in general, I feel like having fixed voltages is nice. That's certainly how I spent all of grad school running on the same cytometer with pretty much the same settings for every single experiment I ever ran. So I do like the fixed setting system, but it's just kind of like, how do you want to determine what your fixed settings are?
That's kind of how we train people on the other platform nowadays anyway, right? It's like we found the best voltage on the Fortessas, and we tell people start with that. If it's too bright, reduce it. Otherwise, you're probably good to go.
Yeah, we have two different. So I did the voltration test on the Fortessas. So we figured out like what's the most optimal voltage where you probably wouldn't want to increase it further than that voltage. So if it's, you can go down, but probably not up from the recommended voltage. But then I've also sat there and run like a maxed out 16 color experiment on our X20, which is really pushing it on an 18 detector cytometer, and fiddled around with like the most precise voltages that I would run using single stain controls. And I always use those settings anytime I use that X20. And I even like have told that to other people for their various experiments. They're like, oh yeah, I like those settings. I'm just going to use those from now on. So different opinions, I guess, as to, but you do make a good point of having a smaller panel. Maybe it's not quite so critical.
Yeah, and I think it's, I mean, I don't know how much this matters, but there is a detector difference as well. So, you know, the Quanteon detectors are not PMTs. They're, I forget what the term they use for it, but it's a
silicon APDs.
yeah, so something like that, right? And that's why they call them gains and not voltages,
right,
etc. So I think the what do you call it? Kind of the standard that was developed for PMTs may not actually hold with the response of these APDs the same way. So for a PMT, I would say that there is a harm in underpowering your PMT. And having too low a voltage can definitely harm things. We've all seen this, right? But having too high of a PMT, like within reason, not like ridiculously high, but within reason, all you're doing is reducing your dynamic range for in the context of a single channel, right? Not in a 18 color panel, but in a single channel, if you put too high of a voltage, you're just reducing your dynamic range. So you're wasting, you know, valuable space to resolve bright things. I don't know exactly how that translates over to APDs. So even like doing a voltration or a gaintration, I guess, on APD, yeah, I've done it for random channels where I thought maybe I could eke out a little bit more dynamic range or more resolution. And, you know, you don't really get exactly the expected profile where, you know, the classic, your voltage is too low and things are moving together and all this, they max out and they start marching up the scale together after that. I don't remember it behaving exactly the same way, but, you know, similar enough that I was okay going with the recommended gains on there. Plus, I think there's, you know, even with the voltration, there is less of a number as opposed to a range. So you could say like, you know, you want to be somewhere between 350 and 425, but probably don't go below 350, and you probably don't want to go above 425, but somewhere in that range you're okay. And then within that range, that's when you can start playing around with, I want to optimize my 16 color panel and move this one a little bit higher, move this one a little bit lower to reduce spread. So I don't know. I mean, I think, you know, you could probably write down some ranges for these various instruments, and then it probably takes a little bit of back and forth for a specific panel to say, I want to fine tune this and dial it in precisely to give me the best quality data. And that might be universal to any instrument, but.
Yeah.
Yeah, and I think that was one of the hot topics at the CYTO, they had the breakout sessions at CYTO, and so they had a couple sessions that were specifically about full spectrum flow cytometry, and somebody had actually done a gaintration test on the Aurora and found that the SciTech assay settings were lower than that. What do you call that point? Like the most sensitive,
whatever. Yeah.
Whatever value, like the best optimal value that you get from a gaintration test.
Yeah.
All the SciTech assay settings were pretty much consistently lower than that. People found that to be interesting. I don't know if that's good or bad.
Is that our segue to talking about the CYTO?
I think so.
Sure.
I'm not keeping track on time, so I don't.
Well, it's interesting that you say that, because one of my problems with that CYTO meeting is at least initially, those breakout groups, they had limited to very small numbers. And it was, you want to go to a breakout group and then it just tells you, well, keep on trying. And if somebody leaves, you can get in. And well, that's, and when we're at a meeting, we all cram in that room that has the best seminar going on. And you may not get a seat, but you can at least hear it. And it does you no good to repeatedly try to get in because you've missed the beginning of it. And a lot of times, especially if there's some talking beforehand, you walk in the middle of something, you don't really know what's going on. So, and I actually had that as something that I talked to, or when we had to write up our evaluation of CYTO, that this was I thought a very inconvenient thing that you couldn't actually get into the groups you wanted to get into.
Mm-hmm.
I think a lot of people shared that thought.
Yeah.
There's definitely.
What was the rationale behind that?
I have no idea.
Like, you're not physically limited by the number of people who are dialing in, are you? I mean, unless they were trying to use like the free Zoom plan to host a meeting or something like that.
I don't think they're really.
I think the meeting was extraordinarily expensive for an online meeting. It was very, very expensive. What were your thoughts? So hard to access rooms. What else? Ryan, what did you think of the meeting?
I watched some videos after the fact of instruments that were launched. I'm assuming they were similar to things that happened during the meeting, but yeah, I did not attend any of it. I
did
contemplated.
You register at all, or just like?
Well, so I contemplated registering.
Okay.
Like, yeah, I'm going to register. I'm going to go, and then I said, you know what, I'm not going to go. I know I'm going to register, and it's just going to, I'm going to get the emails and just ignore them. So I didn't even bother registering.
See, that's the difference between you and me. I'm in the same boat, except I did register.
Okay, so at least they got your money, right?
Missed most of it.
Well, technically, I snuck onto his.
Shh! Don't tell anyone.
I'll edit that out.
Good.
I paid. There you go. Not me, Loyola paid, but I found a couple of very interesting procedures that people talked about in the context of their studies that they were talking about that directly related back to something people in my lab or in my university were doing. And that was kind of nice. I always like it when you can say, hey, they did this, but they've taken what you're doing and they've really made it much more sophisticated. There was one that was tracking of T cells, and it was a very cool procedure. They were doing a little bit of it, but this man had really expanded upon it. And, they've really adapted that in our lab now to track over time and lifetimes of these T cells, which was very cool. So we're doing some of that.
I find it difficult to listen to the really heavily scientific talks in a virtual setting because they take a lot more focus. I feel like for me, as opposed to, I went to the spectral breakout rooms, which are, I guess, you can participate them if you want to. I think maybe that's why they shut down the size. Maybe they were concerned about having 300 people trying to talk at the same time. I don't know if that was the issue. Although having the virtual discussion puts you in a very interesting setup because there's a small group of people who feel comfortable talking on camera, or turning on their microphone and talking, or maybe some people have reasons as to why they can't turn their microphone on. But then you also get the people who are on the chat and typing away furiously. So then you're trying to split your attention between the people who are discussing verbally versus the people who are chatting through text and trying to keep up with both conversations at the same time. And they're sometimes happening on the same topic, and sometimes they diverge into different topics, or maybe one is delayed. It's very hard to get everything.
I think another thing, and this, I think it ties in nicely with, you were discussing before how you're going back to all your videos that you're producing, or you're producing for our website. There's a difference between presenting in front of a crowd and presenting in front of your computer screen, where in general, if I try to explain something to my computer, I have no energy whatsoever. I'm boring and I'm not engaging. If I am in front of a crowd, typically I'm livelier. And it only showed up I think me and Laura were actually watching one of the presentation in the lunchroom at some point, and the presenter was kind of it was a very flatline type of presentation that made it really hard to like want to keep up with whatever was presented, and whatever was presented was already pretty complicated, so you kind of disengaged, let's put it this way.
Yeah, there's definitely an art to pre-recorded videos. I definitely, I can only do the pre-recorded videos at certain times. Like I have to be in the right mood to be enthusiastic, talking to nobody except for my dog. And then even then, I talk a lot slower if there's nobody. You know, you get a little bit nervous when you're presenting to people, even if it's virtually. Like I did a virtual conference last January and I couldn't see everyone, but I knew there was probably like 100 people listening to me, and that makes you a little bit nervous and you talk faster. So I was asked to do a 10-minute talk and I practiced it, I don't know, 5 times, 10 times, and I was on the dot 10 minutes. And then I presented it to the 100 people, and suddenly I was practically done at like 7 minutes because I talked a lot faster when I was nervous. And so when I like pre-record stuff, I actually went through and, I mean, maybe I'm just kind of crazy, but I talk slower and I kind of pause a little bit more. So I actually went and like chopped out all of the pauses so that it would move faster. Or if I listened to it and I felt like the pacing was too slow, then I would go back and re-record it and talk faster.
Just speed it up. Your volume will go higher. You know, you'll be a little like Minnie Mouse.
There's a couple times where I've tried to just make the video faster, and sometimes it worked. But then you get I used to just do all of the videos in one go. So I'd talk for like 20 minutes straight, and I'd be really enthusiastic at the beginning and talking really fast, and then 5 minutes and I'd like slow down. Yeah, that's difficult with how many, they had quite a few pre-recorded videos in CYTO, right?
Yeah.
Yeah.
Yeah, that seems really lame. Like if you're gonna like have virtual talks like this, I mean, why not make them live? At least there's some opportunity for some potential interaction. I guess logistically it's probably easier, but.
So on the ones that I saw that were pre-recorded, the talk was pre-recorded, but they were available afterwards for talking. It wasn't that they weren't there. They just had their whole thing. And sometimes that's nice because I know you guys have seen a lot of glitches when they're trying to run a little video inside their presentation or something. Yeah. And that doesn't happen, and that's nice. So that's the other side of it.
Yeah, I agree with that. We had the GLIIFCA meeting last year, and I edited out the version that's online currently to remove all these. Oh, let me share my screen. Hold on.
Where's the button? Wait, I need a laser pointer. How do I find my laser pointer?
And that even after a year of Zoom, this still happens to many people, including me. I gave a flow basic talks last week. 5 minutes, my sound was off and nobody was telling me. Yeah.
Oops. Well, I was just going to say, GLIIFCA's free this year.
It's free again, yeah. Second year.
Was it free last year?
Yeah, it's the second year online.
Yeah. Hey, nice work on that logo, Laura.
Laura.
Looks pretty sweet.
Thanks.
So I have a confession to make, where it's the 30th anniversary for GLIIFCA this year, and I was at the first one.
Really?
Wow. It was at the Hotel St. Regis in Detroit, and we literally flew in Detroit, and then we took a cab to the place. It was in the middle of downtown, and I don't know downtown Detroit now, but it wasn't beautiful 30 years ago. And we never left the building until we took the cab back to the airport. And it was the first time David Headley wore the Viking hat to dance in. So it was quite a memorable meeting. It was actually really a great meeting because it was the first time we were doing all of this. And you felt, there always was ISAC and stuff, but a lot of people don't go to ISAC. The cost can be prohibitive, you know, because they always packed in the meals and everything. So registration, even back 30 years ago, was $700, which was a quite expensive meeting, especially for scientists. So now we had this local meeting or regional meeting, and it was just exciting to be with people. And there were a lot of people that, you know, Jacob Berger from Cleveland was there. And I've read his papers and done his procedures, but it was the first time to actually interact with him. And that was a very exciting thing. And of course, Howard Shapiro came because he felt that he needed to be part of every flow cytometry society. And he didn't have a booth, but he had a boom box, and he was playing some of his interesting music. So it was a very cool meeting.
Yeah, GLIIFCA meetings online again this year, September 14 and 15.
Nice.
We have 2 days, 4 speakers. The name is not on my mind right now, but you can check our website, gliifca.org, and go check it out.
Check out our new logo and branding.
Check out the new logo and it's all Laura stuff. It's great.
We've been joking that, I mean, cross our fingers, we're hopefully back in person next year. It'd be very sad if we were not back in person, but joking, it'll be like 30+1. Or like a redo of
we're missing our opportunity to have a decent party for 30.
Yeah, it's like the Olympics in 2021, right? We'll still call it GLIIFCA 30, or even though it's a GLIIFCA 31.
I'm going to say this: if for whatever reason, pandemic or other, we're not allowed to do a in-person meeting, either at CYTO or GLIIFCA, I think we should stop doing online stuff, or do it in a different way. Do it like find a different way of presenting the material you want to share. I think packing a full day of events or even a half a day is too much. People are back at work, they don't follow, they don't look at it, and then even if you say, oh, you can go afterwards, nobody really does that. So
we just have to make it like Lollapalooza. We'll host it outdoors.
Yep.
People will come with their COVID test results, fresh COVID test results.
Gimpy outfits.
Yeah.
And we could play music.
And we could play music. What did I miss any hardware? There was I think the Cytec sorter.
Cytec sorter that was announced before the Helios as an upgrade.
Helios, yeah.
Yeah.
Forget.
Yeah, it has a new name, right? Like a
XL. XT? Yeah.
Yeah. Something like that.
I didn't figure out what exactly was different, except everything is a little bit better.
I can't remember.
But no new platforms as far as I can tell.
I wonder if that's just because they knew it wasn't going to be in person and they'd lose some steam
Right.
launching it over the, or if there was like really no development for the past year and a half, so they have nothing to show.
Maybe. I'm kind of curious to hear from vendors what's their experience with online meetings, and like, did people actually go to these vendors' booth and click and start typing hello? Like, I don't know.
So I had a somewhat fruitful demo of the Bigfoot virtually,
Oh,
which I don't know if you guys did that as
Of
well,
course, yeah.
but yeah, I mean, I thought they did a really nice job for a virtual demo. You
Yes.
know, there was multiple camera angles and full view of the software, and Angie kind of walked you through a setup of a protocol and did some sorting like live for you right there.
It's all low tech stuff too. You can see the image, the camera are probably not great, but they show whatever you want to do. So that's why I'm like, whenever you do online stuff, it does not need to be expensive.
it needs to be interesting.
Yes.
It's just, I think people forget that it's a different medium. So
Yeah.
they just think, oh, well, I'm just going to do exactly what I did in person, but I'm just going to be virtual. But you just have to realize that with virtual, you just have to plan things differently and just make it work for that setup and forget all the other stuff you used to do.
So my friend Jill, she's actually the head of an association, and they've been doing virtual meetings, and they do certain things that really kind of make it fun. So they'll have in the evening events that people do at home on these meetings, and it might be everybody gets a recipe, and online we all cook together, and then we sit down and have a glass of wine and just talk about it. And so it's not really about in our case, it wouldn't be about the science, but and you mix people up and you get the chance to, and so maybe some interaction of some sort would be helpful.
Yeah. Another point that was discussed, especially with spectral flow, was the future. Like the spectral flow here to stay, and is the future of flow. And I guess that's this topic I wanted to talk to you about. Like, what do you see the future of flow going forward? I guess like what I've seen at CYTO is like everybody is kind of moving towards the cytomics, or whatever it's called. Like you just look at every single cells and like under all possible technologies. Like look at the genes, look at the expression of RNA, look at the actual proteins, and flow cytometry and sequencing and blah blah blah.
Yeah, I think as a lifelong flow cytometrist, I've gone through bouts of concern for the technology I love, mostly surrounding like these single cell omics platforms, like something like the 10x platform. Their current system, no way. I mean, I would much rather do flow any day of the week. But you can imagine if that technology gets better and faster and cheaper. Their newest 10x's newest box, the controller X or something, and they have a high throughput chips where they're going to actually be able to do closer to 1 million cells per well instead of 10,000 cells per well. Right, so that definitely changes your ability to look at either rare populations within a single sample or multiplexing 10 samples together into a single well. Plus, they have, or companies have kits for doing 40, 50, 100, 200 proteins via the 10x platform as well. So I think you can actually put together, if you wanted to, a 200 parameter, protein via antibody protein analysis, as well as get, thousands of genes read on tens of thousands of cells. So that, I mean, that kind of seems like the ultimate application for doing any kind of single cell phenotyping of any sample, right? The problem is, it takes forever to do these experiments. Sequencing, for me, executing any of these experiments, typically the bottleneck is getting on the sequencer, because, for discovery projects, there's a queue. For clinical projects, they have precedence, as they should. They can get on, whenever they need to. So I might have projects that are sitting for a month or two waiting to get on the sequencer. Sequencers are still expensive. They, are not difficult to run, but still require some know-how. And then really it's just a data analysis and a data processing that, holds something like this back. I say like the kit or panel that the CyTO Fluidigm folks came out with. I haven't tried this, but apparently it's it's a set number of antibodies, and the clustering is built into the kit essentially. So you basically can not know how to do any of this stuff, and theoretically throw your samples through the CyTOF, and on the back end you get beautiful Disney plots. If something like that were to come along for single cell RNA-seq with 200 proteins, I think that would be like a huge game changer. And still, I'd say like flow cytometry would be relegated to the quick and easy applications, or maybe something that has to be read out functionally, like calcium flux or dye dilution or things like that. That's not just antibody based, but dye or probe based. But yeah, in any of my fluorescence based cytometry, I would probably insist on it being spectral, at least having that capability here on out. But yeah,
I would be interested to see how, if in the future there were like these prepackaged type panels, how many people would actually want them? Because in my experience in our core, whenever we try to promote these, like, hey, we have a pre-made immunophenotyping panel, just mix in your blood and run it and it's ready to go. None of our users want that. They all want something very customized. I'm sure there are places where that type of thing is fantastic, and maybe it's more for like MDs who don't do tons of research, and they're just trying to get a sense of their disease and do just generic immunophenotyping, and in that case that's probably a good route to go. But a lot of people that I ended talking to generally want like a very specific panel with very customized markers, and nobody wants anything pre-made. And every single panel is different. So I don't know if it would be different with the 10x genomics platform, if it makes more sense to have, at least for genes, since you're looking at so many genes with that type of stuff, right? Maybe it is.
Yeah.
Maybe it makes sense there, but I'd be interesting to see how researchers want to utilize that or not.
Yeah, and I think, right to that point, the reason why everything has to be customized is because you're inherently biasing your analysis to what you've chosen to look at. If you can throw out the biggest net possible and just run that on every experiment, regardless of whether you need to look at these 10 markers or those 10 markers, all 20 of them are in there every single time, and that was cost efficient, let's say, then you don't have to bias your experiment and put in only this set of markers. You could just say, I'm going to look at everything every single time. Then it could be standardized, I think, and you could get everybody to use essentially the same panel if it were big enough. But yeah, right now, yeah, it's hard to, like, I try to promote at least a backbone. Every mouse experiment is going to have these, 15 markers in them just to delineate all the lineages essentially. And then you can put whatever else you want. Even getting to that point is a battle. But, having the ability to do 20 or 30 colors on the Aurora now makes that easier, because now they're like, okay, well, just as long as you don't take up the FITC and PE channel when you make your backbone, I'm okay. And then they could drop in whatever FITC and PE and APC antibody they want. And that works out pretty well.
Yeah, I think the cost of the data points is what I'm getting concerned about. It feels like it's been going higher and higher with as you move away from flow. Flow is still something that it's actually went down, like with high parameter analysis with spectral flow, the cost of the instruments cut down in half, more or less. Like a Symphony before CyTO showed up was roughly $1 million something if you wanted to get your power machine in your core, and now you got the 5 laser that can look at, was it 45 markers now? You'll have that for like 400,000 roughly for academia.
Yeah.
That was actually my question. Like, what I would like to see from spectral manufacturers would be for them to turn around and help the people who are doing 10 colors and less, and just like help them make experiments on that platform easier. It's kind of a pain if you're just a few markers on spectral. It's easy enough to do, but like it's not as easy as just a traditional flow stometer. So it'd be fun to have them turn around and make their platform a bit easier to handle for these low-end instruments. Like the CyTO has a virtual filter, but you still have to, there's a lot of clicking involved to make that work. So
yeah, I think I could definitely see a future where we have a core facility full of spectral or full spectrum, whatever you want to call it, cytometers. And like that's how the Bigfoot works, where you click new experiment and then it gives you a pop-up window and says, do you want your new experiment to be with compensation or with spectral and mixing? And you choose which route you want to go, and then you continue on. So if we had benchtop analyzers where you just decide from the get-go if you want to go the compensation route and have one color, one detector, or you want to go the unmixing route, then we would have cytometers that do both, and it would cover the people who only want to do 5 colors and maybe they don't want to bother with spectral unmixing, and they can still use the same exact instrument as the people that want to do 30 colors. We're starting to turn the corner where our initial Aurora users were definitely the large panels, and we're starting to get some people go a little bit smaller on the Aurora panels, but they're still relatively large, like 15. But I could imagine we could start seeing people go lower and lower. I guess maybe we do have one that has 4, but it's actually 4 fluorescent proteins. Maybe that doesn't count.
Do any of you use autofluorescence extraction or whatever feature on that?
Oh, yeah. Ryan, we had a full podcast on.
Oh.
One before this one.
Yeah, I watched it.
No, well, it's fine. We'll cut it off.
I just wanted to refresh everybody's memory.
Yes, I've been on a deep dive into autofluorescence extraction because we got a project that was doing human tissue digest. So it was digested liver and digested lung. And the unstained samples on those are absolutely insane as far as autofluorescence goes. And I spent a lot of time trying to figure out how best to manage the autofluorescence. And it's a lot of learning.
I'd say it's a very nice tool to have. It definitely helps with your data. It kind of bridged one of the benefit of using mass cytometry, where you don't need to worry too much about the complexity of cell glass and the high autofluorescence of different subtypes of cells. I don't know if we want to go into mass cytometry. I'm kind of trying to see what's the future of that particular technology, and I've seen a lot of cores where I've seen few cores where they seem to be doing absolutely fantastic. They have tons of users, the usage is going up, and I've seen others where it's just not going all that great. So I don't know. I don't know what's going on with that technology, where it's going.
Yeah, so I don't have a CyTOF at my facility. I don't feel like I'm missing out on anything, especially now that we have. So yeah, I mean, for a while, I was always like, man, I wish I could do 30 parameters or 40 parameters or something like that. Now we can. So I haven't seen, maybe this exists. I just haven't looked too much, but like, and maybe you guys have done it, just like same specificities, 30 parameters, CyTOF, Aurora. What's it look like? Where do you get better resolution? Where do you have problems, etc.? I mean, if that proved to greatly slant in the CyTOF's favor, I mean, maybe that would be interesting to know. But even if it was like slightly better on the CyTOF, I'd still prefer to do fluorescence cytometry, because I have all the reagents. I know how to use the technology. I know all the sample prep tricks and all that kind of stuff, data analysis tricks. So, as long as I can get as much high parameter information as I need, then I'm certainly content sticking with fluorescence systems. Even on, I was also slightly enamored with the what's the tissue imaging?
The Hyperion module.
Yes. So I thought, oh, that would be a great add-on. I mean, maybe I just have a CyTOF that does imaging and not do regular suspension samples. But even there, there's better or maybe just as good fluorescence-based tissue staining instruments, like the one from Akoya called the Kodak system, where I presented in Journal Club a paper from the Nolan lab where they were doing like 35, 40 fluorescent parameters on tissue sections, right? So that's now, and the resolution is like, it has like an actual really good scope on it. So the resolution is just as good as, microscopy. You don't have to worry about the rasterization of, say, like the Hyperion or the other metal ion technologies that are out there. So now I'm like, okay, well, I could do everything I need to do by fluorescence flow cytometry. If I wanted this imaging system, maybe this Akoya bio system is just as good, if not better, than the imaging metal isotope imaging systems. So yeah, I mean, that kind of closes two doors for me to ever really broaching the subject matter of CyTOF again. So I mean, it kind of seems like a dead end to me, at least. I don't know. Like you said, David, there's definitely groups that are very successful with the platform. Kudos to them, but yeah, I don't see it on my end.
Well, from an economic standpoint, at a smaller facility like I have, it's really hard to support a CyTOF.
Yeah.
You really have to have a critical mass in order to make that something, because you need dedicated personnel and room and blah, blah, blah. And obviously it's much easier to get a SciTech in your lab because people are more comfortable and you can use it for more. You only want to do expensive big experiments on the CyTOF. You don't want to check your CD4s or whatever, you know.
Yeah.
So
Yeah.
I'm not going to use it for a 10 marker experiment.
right.
Yeah. The Cytec runs on DI water. The Mass Cytometer works with an argon tank, which is basically like we get them $700 at Eward, and that's going to last 2 weeks.
Oh,
10 days.
10 days.
and then the heavy metal antibodies, those are expensive, you know. So
I thought last time I checked, they weren't actually that much of a price difference. Maybe
really?
some of them were a little, but I thought they were roughly, well, there's such a range with flow cytometry antibodies. You know, you can get like a CD4 FITSI for $40.
Yeah.
Or you can get some of the BUV dyes for $500, right? So there's a big range, and I think there's less of a range in the metals, but I think most of the metal antibodies that you can purchase are somewhere around $200-300, if I recall correctly, which is.
But the issue is that you have to duplicate your antibodies for if you're going to do fluorescent-based assays or mass cytometry. Which is why I'm looking for the DNA-based fluorophores, Phytonex, and so on to start being available for sequencing. That's going to be fun.
That would be cool.
We will see.
I don't know. I just can't figure out how to convince people to use the CyTOF. I mean, I'm not a salesperson. I'm terrible. I tried in college to be a salesperson at Nordstrom, and it is not the job for me.
Can I say this? You have a way of presenting things where you're going to focus on the worst aspect of the technology first, and then turn around and say, well, look, it could work.
Probably kind of. Yeah, I probably do that. I like to just give people the facts.
Yeah.
But yeah, I'm a terrible salesperson. So, you know, I try to give people an unbiased, here's what happens with the CyTOF and here's what happens with the Aurora. And they both have their pros and cons. Maybe I talk about the cons a bit more. Maybe I should focus on the pros a bit more. But I don't know. After talking about both, everyone just gets scared of the CyTOF, and
yeah,
they don't. I think it's mainly a cost thing. My sense is it's a combination between cost of buying reagents that are very specific to this one single experiment that you can't use anywhere else for the most part. I mean, I guess you could use their water and PBS for whatever you wanted. And just the fact that it's new and different and seems unfamiliar and scary.
Yeah, the new technologies are hard for people to get on board with from the get-go, like whatever you do.
So that's probably why the large facilities that have this whole, you know, there's 30 PIs at a university that are using the CyTOF, then it's not really new and scary anymore to anybody at that university. So they have that extra momentum that we just never quite got.
Yeah. Last point I wanted to bring tonight, and still on the future of flow, but like I wanted to tie everything with a nice bow and back to Pat's point initially, which was she was trying to hire people, and I feel like the job is changing fairly drastically these days. Like we get instruments that are at the same time easier to use, but can do pretty complex stuff. So the Quanteon, you don't really need somebody to like fix the voltage around anymore. Like this is pretty much given now, but then you get to like in our core, Ryan and I would hire entry-level position people and stick these people on our cell sorters where they would do fairly basic stuff like GFP sorts or a few colors. And but now we're getting a Bigfoot in our facility. Suddenly you're moving from like your GFP few experiments, and then you're going to be facing people that are bringing like 24 markers in their assay, and you need to do spectral flow, and potentially they might want to do a V-SNE transformation and then gate on that and sort on these cells, and then it's pretty rough on a newcomer, right?
I feel like that even translates to the students. I've been talking with some of the grad students that I help with the spectral plots. And, when I was the very first real flow cytometry experiment I did with 6 colors, and I thought that that was kind of on the harder side at the time over a decade ago. But now you've got these like second year grad students who are handed like, oh, the student before you did a 25 marker panel, so continue that on. So here's your first big experiment as a second year grad student, and you have 25 markers. Good luck.
I wonder if that's always the case though. Like everybody always thinks like what they're doing today is so complicated, and then fast forward 5-10 years, you're like, oh, that was so easy. Now what we're doing today is really complicated. I mean, I get what you're saying, David. I mean, it's not like, it definitely seems objectively more difficult for somebody who's very new or has no experience in flow to kind of just jump in and go from no flow experience to, spectral unmixing on the Bigfoot, sorting into 384 well plates or something like that. But I think also the tools to accomplish these things are so much better than they were, even a few years ago, that it's not, I mean, yeah, it requires a lot of training and hand holding and whatnot, but it's not unthinkable that you could have somebody relatively new who could accomplish these things. Maybe they don't understand everything that's going on, but they can go through the steps and have a successful sort. Just like, back in the day when we were getting new people in and throwing them on the MoFlow and like, oh, you have to fiddle with all these, knobs and whatnot on this big rack of electronics. Spend a few weeks with them doing this day in and day out, and they get it. I mean, they don't understand what any of these, moving these BNC cables around to threshold on different channels means, but they can do it and they can get a sort done. So, I mean, all that to say is, yeah, I agree. And I also think that, that the tools becoming easier to use make this a possibility, and that we could still, go down this route of hiring entry-level people and getting them, up and sorting at least proficiently. I always thought maybe 6 months was a good breaking in point to get somebody relatively competent on a sorter. But then it's like, yeah, nothing beats experience beyond that. Everything that you run into is potentially something brand new that you've never seen before. But then next time it comes by, you're ready for it. But the other thing I would say is, I do think though that a new person with a year of experience in a core facility is pretty darn valuable in terms of skill sets that they know. Like if you can imagine you brought somebody in who's brand new and they came out a year later proficient in running the Bigfoot, like that person could demand a pretty good salary anywhere else, which again gets back to kind of the HR issues of holding on to these people so that they don't leave for the big pharma who's trying to lure them away with lots of money. But yeah, I mean, I would say like if I had somebody who knew how to run a spectral analyzer and a sorter, like I would hire them immediately. So there's that part of it too.
Yeah, I guess my point there is more that the job changes. You're not so the MoFlow, for example, was so complicated that you had your dedicated instrument and you would probably not do much else than follow the sort. And with the newer sorters, you're kind of, you have some freedom to explore some other applications, some other things you can do in your lab, develop some new skills, and I guess that's where when you manage your core, when you manage your team, you need to find some avenues for all these efforts to make it useful, and just not waste all these hours of people have in the day.
Is that a good thing or a bad thing?
I think it's a really good thing if you do it properly, and otherwise it's just a giant waste of time for your staff.
Yeah, I guess it's different for the core facility staff versus the grad students. I can see Ryan's point of you just now have to do different things. So maybe you didn't have to spend your time fiddling with knobs and settings, and like now with the newer technology, all of that is set up. So now you have to spend more of your time, I don't know, like dealing with all of the controls for your 20 marker experiment and making sure that you set the gates properly to get the spectral and mixing to work. And like there's different things to think about, but timing wise, you're still spending time doing something. But for the grad students and the postdocs who now have to come into this 20 marker, 30 marker panel, it's tough for them because now as the technology advances, you can get data faster. And so, you know, you're looking back at like publications from 20, 30 years ago, however long, each like figure one has one panel. But nowadays, figure one has like 20 panels in it. And grad students and postdocs are expected to do all of that. You're expected to do the 30 color flow experiment and the single cell genomics and the, you know, this and that and this, and have the big data and be able to analyze all of that, which like people have bioinformatics PhDs, but now immunology PhDs are supposed to learn how to do R, and like there's just so many more things to do now for people who are publishing, and I feel bad for them. So on that end, there's a lot more to deal with, but I guess just as far as operating, it's probably the same work, but just different focus.
Yeah.
And not from early days. From early days, it's much, much different. I mean, the data was very simple in the early days, but if for you to get, I had an instrument called an IFA, which was an improved FACS analyzer. It didn't have a laser. It had a cadmium doped mercury arc lamp. And then you would put different filters blocks in front of it to get different excitation wavelengths. And to get that all in, I mean, it took me the first 7 months just to get a side scatter signal on that instrument. I mean, so I would just run forward scatter as a histogram. And that's how I would do my gating initially, because you couldn't, I mean, it was almost impossible to do. So you would spend hours and hours and hours just trying to get the, and they weren't stable. And so it was a real art just to get something that looked good off of the instrument. It took a long time to do that.
Right.
So there is a huge, I mean, what I see from when I started to now is incredible. Yeah.
All right. It's been about an hour, I think.
Or two.
Anybody wants to bring a new topic, or we can close this for tonight? Thanks, Sue, so much for your time. And
Thank you, Dave.
in lieu of this 1 year or so anniversary, I want to thank every single guest we had on our podcast so far. It's been great to have you guys in and talk about your experience. For YouTube listeners, you need to like and do stuff. I don't know. You'll get an email on what you need to do.
Subscribe.
Do
Hit the
the YouTube
bell.
thing where you point below.
You can find our other videos right here.
Yeah. All right. Thanks so much, everybody. And
Thank
we
you.
will see you soon.
Thank you.
Bye-bye.
Bye.